سد الباب لو سمحت والكلام. يعافي عليكم. هاي أول بالبداية أول سنة. شو راح تسوون بعدين؟ على كل واحد يقدر ينتقم من عندكم، اللي غبتوا فيها يجيب سؤال فيها يعني بين الموضوع. Today our lecture is related to neurology. Not a lot of the physics in there.
So we finished the heat, the chapter?
They finished the heat, yeah. We start a new chapter, which is related to the electrical phenomena in the body.
How many chapters with us implemented?
والله ما أعرف.
So I should ask Waheeb tomorrow.
أنا وهيب. Send him email, see it all.
Okay, thank you.
Now, before I start, I asked Waheeb this morning, what does these red things mean and what does this black, no, green things mean on the things, okay? So I will read what his answer is, and to be honest with you. معناها أنها ليس الأحمر يقصد يعني. معناها أنه ليس من المطلوب اتقانها وإنما يتم الأخذ بالمضمون. I don't know what does that mean. هي للعلم فقط.
نفهمها بس مو
ما تفهمها يعني ما مطلوب الامتحان تقريبا. أوكي. وليس للدراسة والتقييم. أوكي. أما اللون الأخضر فهو عكس ذلك، فهي تعني أنها معلومة تستحق الفهم. أوكي. مفهوم؟ نعم. الزرقاء هم يعني مسموح فيه، يعني لازم تفتهموها. احنا الريد وجرين وبلو، ما عدا الريد البقيه كلها لازم تفتهموها ومطلوب للامتحان. Okay, we say electrical phenomena. Let's set this. Now I am walking now, isn't it? Yeah. How I can walk? Well, my feet. But how I use my feet,
moving.
Push the ground, after that the ground pushes so you can walk.
Oh, that is physics, I'm talking about neurology. Huh? Isn't it? إشارات الأحمر للفهم فقط، وغير الأحمر شلون؟ ريد ستار؟ موجود المحاضرات ما بتشوف فيها علامات حمراء وعلامات زرقاء وعلامات خضراء. فالحمراء فقط للفهم، مو للتقييم. البقية ألوان أخرى للتقييم للامتحان. Okay, I say that how I walk. Yes, actually, my brain sends signals through the neuron cells and tell me walk. Or my heart is working because it's all electrical signals produced in the body. Okay? Now, of course, if you look at the electricity or the same in the human body everywhere, of course, that is electricity conducted by the type of the cell we call it a neural cell. Neural cell, okay? It's excitable cell. I can excite it, means that you can change the electricity, producing electricity in the nervous system. So it is an electrical signal is needed for nervous system to send signals through to the brain and making us what? Possible to walk, think, and study, I would say, or running out of the class after scanning our face. Okay? So electricity in the body need is for what? Why electricity need for? For nerve impulse conduction. There are billions of the nerves in the body. So need connections between this nerve telling this one, moving that one, going to the brain responding through nerves, and so on. Okay, so that is the kind of the thing is about that. And sensory information acquisition and processing. I see you. Okay, so all go to my things, my whatever in the retina sensors, send information. That is through the electrical signals. Actually, we call it action potentials. And then it's go to the brain, and brain interpreted things, and that is your shape telling me you are Fatima or Zainab or Leila. Okay? Of course, the same thing is happening with the hearing also. Okay? When you have hearing, you have the sensors that is kind of the brush going hair cells inside the hair, touching is these things with the different electrode producing the signal, and signal going to the brain, and then brain transmitting the electrical signal to the talk, to the words. Electrochemical activities electrical signals can produce some kind chemicals, and these chemicals can be recognized by another cell and then converted again to the electrical signals, especially when you have gaps between the nerves. Okay? And also excitation condition of the heart, so exactly that is how is the heart beating, things. Next lecture we go to see that is how cardiology measure what type of the voltages in the heart, telling us the heartbeat, okay, how many beats per minute, and so on. Okay, now in the body, so basically that the cells consist what is core, isn't it? Yeah, and you have a liquid outside and a liquid inside. Okay, inside body they are responsible for the conducting electricity. Say for example, sodium, potassium, and so on. Okay, different chlor, and there's all kind of the materials in here. So you have a calcium, magnesium, and potassium and sodium have the specific electric charge. They are ionic. They are, most of them, they are the chlor. The other is all positive charges. It's all positive charges. Of course, that is the inside the you have inside the cell or outside the cell, or the cell is protected from environment by the thin tissue, we call it membrane. Okay? And this membrane of the lipid on it, and it's a type of the lipid, type of the protein, I see. You see the lipid, and this is to create barrier that only contain substances can reach to the cell interiors. Not only does this environment. So they have a membrane now. You have a membrane protecting cell from outside environment. Okay, this membrane is type of the protein. Let say for example water going through diffusion, and also inside also they have some kind of the proteins. Let the potassium going in, potassium going out, and sodium coming into the cell. This is producing some kind of imbalance in the cell charge arrangement and producing electricity. Okay? So we say that the content of the cell is protected from the outside environment by the cell membrane, and this cell membrane is made of the protein, this type of the protein, lipid, could be 500, 600 types of the proteins exist in it. And that created a barrier only to certain substances to cross to the cell interior. Not only does the cell membrane also can produce some kind of the electrical current. We call it membrane potential. And this membrane potential is coming from, say for example, differences in the charge of the concentration outside and inside the cell. So it's if you have, say, for charges outside and charge here, if they are balances. The balance and producing electricity. This is producing electricity. This type we call it a resting potential. Okay, resting potential is a potential which is really nearly 80 or 70 millivolt, minus 70 millivolt. Okay, so they will say that the resting cells are negatively charged inside. So you have charges inside, they are negative, and they are outside, they are positively charged. And due to the slight differences in the concentration, producing what? Electric field. We call it that membrane potential. Okay? This is at rest. And the membrane potential at rest is nearly about 70 millivolts, minus 70 millivolts. Of course, that is maybe happening before any flow is happening between them. But also there is two types of the flow. One of them is diffusion, which is charges going from high concentration to the low concentration, and the other one's through the channels. As I said, sitting on the membrane type of the protein, this type of the protein let potassium going in, and we call it potassium gates, and then other signals which is let sodium going in, we call it what? Sodium gates or channels. Okay? They go only through these things. And later on I tell you how they open and they how close, and it's what is the action potential is. Okay? Okay. So let's say that controlled by flow of with the proteins that are sitting on the cell surface. Okay? And what is the proteins are called ion channels or gates. Okay? And when they're related, this allows positive or negative charge moving relatively. Okay? Okay, but when we say that this gate is open now in general, and then positive charges move to one side, right? You're producing more signals. Okay, it's become more positive. When you have more positive, more charges involved, higher signals. This type of the signals we call it action potentials. Okay, so you have two types of the potentials. One of them membrane potential. Okay, and the other one is action potentials. They are different. Membrane's potential is not dependent on the charge. It doesn't care that is it a charge positive or negative. It depends on the concentrations. Higher concentration, low concentration. Okay, and a result in producing electric field. And action potential is dependent on the charge. Okay, we come back later on to see those. Okay, if it is a picture of the neuron cells, mainly consists of the four parts. Okay, the first part is called dendrite. Okay, this is like tree shape of it. It's branching. This has thin structure which is branching out. Okay, so one branch and another branch and multi branches is happening here. Actually, dendrite is a Greek word, means what you call الشجرة شو اسمه الشجيرات الطرفية أو بين الشجرة والفروع. Okay, so that's a dendrite. And then you have a cell itself here. Okay, and you have, that is the second part, and then you have axon, which is transmitting the signals producing in the cell to the any part of the world, and the any part of the body. And then what is happening about axon terminals in here. This is axon terminal is the synapse, they call it synapse. It's a small gap between this axon terminals and then dendrites of the next cell somewhere here. Okay, so I had a connection one to the others through synapse. On the synapse, I'll tell you how later on how you can spin. This is actually that is if you look at another cell, this part of here is coming in here. So these things will be really behave as the receptor and the receiver and the, what you call, the sending and receiving the signals. Okay? Now, of course, that is, I will say, a typical neuron process of the cell body generate الشجيرات الطرفية وأكسون عربي الساق, and then synapse is the gap between two neurons. And I say that the dendrites is a thin structure. This arises from the cell body, often extending hundreds of the micron. Actually, that is the longest neuron is in the body, is the one is starting from the spine going to the toe. Okay, some people will say sciatic. Some cases you have somebody getting pain from back or going down all the leg. That's the problem with this nerve here. Okay, and then I think in Arabic they call it Naser. Okay, so that is the sciatic and sciatic nervous. So this is the longest one, depending on the tall of the person, and probably 1 meter and more. Okay, so this is the, but it would be very thin also, is micrometers in there. An axon is a special cellular extension that arises from the cell itself, and then its main thing is to be what? To transmit and receive the signals. Of course, that is, ions cross the neural cell's membrane. I just ignoring that thing. Can cross neuron cell membrane. We say that is a membrane under 3 influences. Okay? I say, first of all, that we say membrane permeability, and then have membrane permeability. I said that is type of the proton is lipid sitting on the thing and make a gate for the potassium and make a gate for sodium. Okay? So that is the opening pores yani holes, okay, or gates or channels. Okay? The other one is what be diffusion. I say that as diffusion is happening going from one high concentration to the low concentrations. Okay, that is the member, we learned that from school. Okay, and electric field and the potential field, actually this is because they are charged. So the electric field is playing a part also to push the electrons or ions to go from one place to another place. So 3 things in happening when the, say for example, ion is crossing the membrane. Now, briefly that one, we come back to it, how action potential is produced. Okay? Now we say that is, remember that I said this is the synapse between these two, and then we go on the dendrites getting the signal, and this signal, these signals make the stimulate the, example, sodium or potassium here. Okay? But we say that in general, triggered signals is coming in here. Membrane permeability, pores with the membranous wall. Let us make a pores, means that opening the channels to let the potassium and the sodium going through. And the diffusion of the sodium and potassium, of course, that is, they have different gates. Okay? So there's 2 باب موجود هنا، واحد للصوديوم واحد للبوتاسيوم. Okay? So it depends on the concentration that is on the membrane, and the charge differences across the membrane producing some kind of the electric field, and this electric field can be transmitted through the body to a proper place which needed to. Of course, the other thing would be diffusion, we said. And a diffusion is happening from high concentration to the low concentration. So I suppose I have some kind of the dye in here. Okay, and we have a water floating on the water itself. Yeah. Concentration is in here compared to here, which you see that is nothing is happening. So nothing in here. So it's happening that the those dye molecules can diffuse to other part of the tissue. Now, this will continue until you're getting equilibrium. So the number of the, say, molecules you have in here is equal to number of molecules on the other side. Okay, that is the diffusion. It's happening between two materials also. The membrane-related type of the molecules go in this direction, but doesn't let you go to the other direction. So one molecule is going to this direction, the other one is going to other molecule directions. This is continue until you're getting what? Equilibrium again. So that is the case in here we're telling you. This is two types of the molecules in here. And then it's about diffusion. The what you call brown is coming down, and yellow is going to this direction until you get in equilibrium. Okay? And the property of simple diffusion is a spontaneous movement. There is no control. There have no certain paths. You have to go to this direction, or you just go to that direction until you go to that. So there is no. It's always moving from the high concentration to the low concentrations. And it is occurred in the radium kinetic movement, no special path, as I said, okay? And does not need energy. There is no energy to put them back, So three properties of the spontaneous particle, of the high concentration, of the low concentration, does not require energy. And the ion movement. And this is continuous until the getting what? And again, there's a lot of the things are diffusion across the membrane again. And you say, for example, I have two different types of the molecules in here. If the color is diffusion from A going to B, and also the B go to the A. So that is the number concentration is here is different. You see, that is higher concentration in this part compared to that part in here. So we have net diffusion. When you have membrane potential in here. Okay? And this is continue until you're getting both of them in equilibrium in here. If you look at that one in here, this is also this molecule is going from area A to the area B and from B to the A. But we say that the number of the molecules in this area here is equal to the number of the molecules in that area. So there is no diffusion in there. In this case, that we don't have electrical signals in here. Okay? Now, of course, that when you're talking about sodium and potassium, they are all an electrolyte, and they are in water, okay, or some kind of the fluid outside and inside the shell. So the electric property of the plasma is an equal distribution of few ions of sodium between the outside fluid and inside fluid, okay, and you have the selective movement as we go through the plasma membrane. As we said, that there's few things in here as well. Okay? Now, in normal, when you have the, say for example, potassium outside the cell, sodium outside the cell and potassium inside the cell, usually you have more negative charges inside compared to the outside. Then we call this one is the resting position, okay? So the membrane's potential characterized by excess of the positive charges and outside from the cell. When you have a resting, if I change the concentration of the outside relative to the inside, or inside to the relative to the outside, then I have what? Electrical potentials. And that is just telling you that is what is the total volume of the things. Well, actually, that is not. And they tell you that is how the plasma, what type of the materials in here. I just for look at it, I think. We said something, a lot of the students, they make confused about cation and ions, okay? We see that is that if you have an atom here, neutral atoms, they're always number of the electrons equal to number of the protons. But this atom may be losing electron or gaining electron. When losing electron means that is the half number of the protons is increased. So I have a cation, the positive charge. When you have, say, for example, gaining electron, a number of the electrons is increased, so I have an anion. Okay? So this is the thing. If you look at that, this is cation, because what is the number of the protons is about 5 in here, or just 3 outside in here. So this is positive, and this is negative, because the number of the electrons is higher than number of the here. Okay? So basically, it depends on the you gaining electron or losing electron. You cannot approach the nucleus itself. If you got to electron inside proton or the things, maybe you end up with making a bomb. Okay, it's also again membrane potential. If you look at this here, if you count number of positive or negative charges in here, they are all equal in here. Then when they are equal, no potential between them. It's no potential, okay? And if you look at the membrane, if you look at this side in here, look at it, they have number in that charge is higher than number of the side charge. Number of the charge on this side is higher than number of the charge on the other side. Then you have what? A potential. That is, there are a charge, but there is no potential because there is no differences in the number of. Yeah. And they always say you have a potential because there is different numbers of the charges in there. Okay? Of course, that basically that you have a charge of the things. So not all the charges they are contributed. That they all close. So that is the charges responsible for the producing with the electric field. So not all the charges involved in here. So they're going to have a positive and negative attracting to each other some kind of the barrier, and this region is producing what? Electric potential in here. Okay? Of course, the equilibrium potential can be calculated by, or membrane potential can be calculated by the nearest equation. Okay? Is this equation in here. And if you look at this unknown here, or probably that you have to familiarize yourself with the log and ln, what is the difference between them. Or probably you don't need to just learn how you're using calculators, okay, to find out what is the differences between them. But he explained what is the differences between them. If you look at this equation in here, the equilibrium potential can be, say, from R, which is gas constant, T is a temperature, and Z is the ionic valence. That is all constants we have to get on. And F is a Faraday electric constant. And there will be this concentration of the ion outside the cell and the concentration of the ions inside the cell. Quite easy if you have only one type of the ion. Say, for example, you have only potassium, you can work out this for potassium. How many potassium outside the cell, how many potassium inside the cell. Okay? Now, of course, that you can calculate these things here at the 37-degree or room temperature. You get this kind of the constant. And you can use that one only when you dealing with the temperature of the 37 or 37. Okay? I cannot remember properly. Just true for the certain all temperatures in here. Okay, again, just talk, and I say the rest. They have the potassium, sodium outside the cell and inside the cell potassium, and then one of them is higher than the other one, producing some kind of the potential reading in minus. Okay? Okay. Now, of course, that is quite easy to, if you have one element, we call Nernst equation. But if it becomes more complicated, then you have the Goldman equation in here. This Goldman equation, you put that as different materials. Say you have potassium, sodium, and chlor. And then also the permeability of the membrane also go to the calculations here. Okay? Now this is the equation we say permeability of the potassium inside the cell, permeability of the sodium. It seems to be that the membrane is not permeability equally to all ions. They are different. Okay? And chlor also that is, and outside. See that is both of them in, the other one is out. Okay? And also potassium permeability out, sodium out, and chlor in. So that is, you can put all the things in the example you have. At the temperature 37, that would say that 37 probably, that's 37, that was all constant, has become 61.5 millivolt. And this is the equation in here. And this is the table I have in here. So I have a potassium outside extracellular concentration, outside the things, is about 10 millimole per liter. And inside is about 150 milliliters, and then permeability 1. And sodium 140 outside, and inside liquid is about 15, and permeability 0.04, and permeability 0.4. From this table, you can see that the permeability of the sodium is less than all the other ions in here. Okay, so you plug all these equations in here. At the end, you're getting about nearly minus 52 millivolt. Okay, I'm just telling us that is the resting condition is some kind of the voltage in minus millivolts in here. Okay, I will leave these things in here. You can read that one here because I will going to summarize this on this drawing. If this one is not in your slides, okay? So basically happening in here, okay? I have in resting condition. Remember that you said a membrane, they have a gates, isn't it, yeah? Sometimes you're prone to let the potassium going out and then sodium going in, okay? Now, in the resting condition, there is no diffuse of the sodium to the inside and then potassium from inside to the outside. So the gates are closed. The gates are closed. If you look at this one, is the picture in here. So the sodium is outside here, and then the potassium inside. If that one is sodium gate, then you can see closed in here and closed in here. So I am nearly at this position here. Okay? And I say that is resting position. Okay? Now, as I say that the signals through dendrites coming in stimulate the sodium gates to open. Okay? So we're going to see that, as you see, when this gate is open, all sodium is coming into this side in here. Okay? Of course, that is not going to be threshold of the needed, threshold of the power of the voltage needed in here. So not only have to be voltage is enough to promote this from 70 to the 55 things, nearly 55, nearly 25 millivolts difference. This in this position, that is the threshold of the things. You remember that is they have polarized. We have in the beginning, and we could look at the polarized. I have a sodium here and the potassium in here. They are polarized. You know what's polarized? All girls in this side in here and boys in other side is polarized. Positive charges and negative charges. Okay? So this is a polarized one. Okay? When the number of the sodium is increased coming in there, you're getting some depolarized. Okay? There is no more polarized in here, so depolarized. So in that case, because the number of the sodium is increased, then you have action potential is increases. Okay? Now they say that is that you have there's a region of the what? Depolarization. This is depolarization in here, which is the gate is open and a sodium in here, number of the charges is increases, you're getting depolarization until you to shooting out, so the maximum ones. Okay? So they get a maximum near the 30 millivolts is produced in here. It's a huge, isn't it? Yeah? 30 millivolts in here. Now, after that, sodium increases there. This sodium sends the signal to the potassium gates to open. And starting, you see that this thing, this opening in here, and this round, small round thing is coming to the thing in nearly the closed. Not closed, but the gate for sodium is nearly closed, but there is not letting any sodium coming in more. Okay? So in this case, that is the potassium gate is open and the potassium is going out in this direction in here. Okay? At this region, we say that repolarizations. Okay? Be repolarized, depolarized, repolarized. This is a process going to the polarized again. Okay? We got depolarized in this region in here. Because this opening and closing for potassium is very slow, so the voltage is go down beyond the 70. And this we call it the hyperpolarizations or hyperpolarized. Okay? And at the time when you go down, both of them getting closed again, and then we come back to the resting condition. And this process happening every millisecond. Okay? That will get one pulse and another pulse and another pulse. Okay? They are very fast. Actually, that is, I was put in my mind 100 kilometers, not right, but it's this huge number, kilometers per hour traveling. Okay? I'm going to repeat that one for you. Okay? Now, a resting condition, I have some kind of the differences in the number of the potassium molecules and number of sodium molecules in there, or sodium ions, sorry, that's not molecules, sodium ions and the potassium ions in here. Of course, that is the signal is coming, producing, how we producing potential action. This would be when you have at a resting condition, the potential is about, some of them say minus 70, minus 75, minus 80. Okay, so that's what the important for us, that is starting more than 70, okay, or 70. Actually, that is in 55 is not have to be put in. Now, that is the resting condition. Now stimulates the signal coming in, triggering signals coming in here. Make what? The sodium gate is open. Okay? Protein let the sodium going through the type of the protein. So what is happening that is, because they are polarized, more sodium is going in here, so you go in depolarized. Okay? Depolarization. Then you have depolarization, means that you have number of the sodium increases in here, then you're producing more action potential or more energy or more voltage in here. And this multi-voltage is growing up by increasing the number of the sodium is coming down in here until you're getting some kind of the saturating point, which is the maximum voltage we get. Okay? When you're getting that, the sodium signals, send sodium ions send signals to the gate in here to let the gate for potassium is open. So this potassium is open here, and so potassium ions are leaking out to the outer fluid in here. Okay? And this process, we call it what? Repolarizations. Okay? But I'll say that is because this gate is very slow, so the things is going below the 90-70. We call it this one a hyperpolarization, and after a while it's back to the resting positions. So basically, we have resting position, depolarization, which is positive pulses, okay, and then repolarization and hyperpolarization and back to the rest. And these things will be repeated very frequently, and that is the transferring the signal in here. And actually, that is, I will let this also go because I was just explain here. I mean, if you look at the bottom in here, I have a link in there, so you can take this link to the Google and get more explanation and then more details. And actually, this one is the same thing as how the potassium is coming in, how the sodium is, how this is. Let me say that as I go quickly in this. This is the position. I have side and then the potassium inside in here. And then the gate is open, sodium is coming in. So stimulase initiate local depolarization. A stimulase changes the permeability of the patch of the membrane, and the sodium ion is diffused rapidly into things. Actually, that is all those things you don't need to say. I just will with the gate it opening, other things. Okay? And we'll still continue, and then a depolarization is happening because that is depolarization generation of an action potential. Okay? If you do multiple choices in the thing, in hand. Yeah, if there's somebody asks you which one is corresponding to the action potential, polarization, depolarization, repolarization, or hyperpolarization. Depolarization. Okay, that is the way you think about the multiple choices. Okay. So that is all explain things for now. The last part of the neural cell is the synapse. The synapse, that say, for example, if you look at this picture in here, this is axon terminal. Okay, this is one of the branches of the axon terminals in it. Okay, and you have the here neurotransmitter receptor on the top of the what? Dendrons. Okay, so I take two of them and let's say it's now nerve impulses coming from the axon. Okay, this nerve impulses when you're getting close to down here, make some kind of the chemicals coming out of there, and these chemicals will be sensed by the other nerve and producing pulses in here. So that is the way going to the atmosphere. So let me say that topic gap is that separate terminus bottom of the one neurons from the receptors usually located at the dendrites of the another neuron. When neurons communicate, they release some kind of the chemicals that must travel across the gap, and then we can stimulate the what you call the nerve impulses or action potentials in here. So that is the way how you from one neural cell signals go into other different neurons. So that is the picture we gave you, that is neural cells. That is only one type of the neural cells. There are about 4 or 5 types of the neural cells in there. Okay? That is my neurology lecture. Okay. Of course, that we say impulses are below cross the synapses to solve. Neurotransmitters are released from the nerve action terminals and then dendrites pick it up and producing actional potentials in the end. Okay, just more explanations or more complications in there. Okay, and I think we end up with the lectures. So any questions, anything? And yes, yeah. Say it again.
Does it 61.5 consonant in nearest equation?
And then this would be constant for the certain temperatures. And gas constants, all the things that they put in. Okay? If they give you, say, temperature, all the, say, R, T, Z, F, all kind of things, and whatever those related to, then you have to use the same equation. If they say that at certain temperature, then you go to 0.65. Okay? Any other? From previous lecture? Nothing. Okay, we'll assume you understood then.